Hello, everyone. Welcome. My name is Jennifer Nyman. I'm a senior principal at GeoSyntech Consultants, the coordinator of the webinar series on behalf of SERDP and ESTCP. I will be facilitating today's event. Today's webinar focuses on DoD-funded research efforts to develop treatment technologies for munitions and wastewaters. First, Dr. Jennifer Vedas from the University of Utah will describe methods for selecting efficient, cost-effective, and sustainable treatment trains for mixed munitions wastewater. Jennifer's presentation will be followed by a brief question and answer session. Second, Mr. Brian Hubbard of the Joint Program Executive Office, Armaments and Ammunition, will discuss field demonstrations of an integrated nanoscale zero-valent iron hydrogen peroxide process for destruction of munitions compounds in wastewater. His presentation will also be followed by a brief question and answer session. And then we will conclude today's webinar with a longer Q&A session featuring both of today's speakers. The next few slides provide some instructions on optimizing your webinar experience. If you haven't already done so, you can download Zoom at the link shown here. If you cannot download Zoom, you can also view the slides using a compatible internet browser and by creating a free Zoom account. If you are unable to view the slides on your screen, or if your screen freezes, you can try keying Control with F5, and that will perform a hard refresh. For audio issues, if you are accessing the audio through your computer, you can click the arrow next to the Join Audio button and select Test Speaker and Microphone, and then follow the prompts. If you continue to experience difficulties, please call into the conference line shown here. At any time, you may also submit a comment using the chat box for any technical difficulties, but please use the chat box just for comments related to difficulties with technical issues. The Q&A box on the Zoom screen should be for questions for the speakers. Finally, if you continue to have any difficulties, you can always download a PDF of the slides. It's already uploaded to the link shown here. And then you can call into the conference line, which was provided to you in your webinar registration email. Finally, we will also be live streaming the webinar on the SERDP and ESTCP YouTube channel at the link shown here. Today's broadcast is listen only. You may submit questions by using the Q&A box on your screen. You don't have to wait until the Q&A session to submit your questions, and it would be great if you submitted questions in advance of that session. When you do send in a question, please add your name. Please add the name of your organization at the end of the question so we can identify you. And with that, I would like to introduce Dr. Andrea Leeson, who is the Deputy Director of SERDP and ESTCP, as well as the Program Manager for the Environmental Restoration Program area. She's been with SERDP and ESTCP since 2001. Before that, she was a research leader at the Battelle Memorial Institute, where she conducted scientific research on in situ bioremediation and the design and implementation of innovative biological, chemical, and physical treatment technologies for site remediation and industrial wastewater. Andrea received her doctoral degree in environmental engineering from the Johns Hopkins University. Andrea, please proceed.
Thanks, Jennifer, and welcome to everyone who joined us today. Before I turn you over to our speakers today, I'd just like to give a little bit of an overview of who we are. I think many of you probably are familiar with us, so I will not go through this in tremendous detail, but at least to give you a little bit of an idea about the programs. We represent the DoD's environmental technology programs, and these are two companion programs, which is SERDP, the Strategic Environmental Research and Development Program, and ESTCP, the Environmental Security Technology Certification Program. SERDP is our traditional science and technology program, or an R&D program that was established in 1991 and is a partnership between the DoD, DOE, and the EPA. And essentially what we are doing in this program is we want to take fundamental to applied research and use it to impact real-world environmental management. ESTCP is the demonstration and validation program, and we work very closely between these two programs. Under ESTCP, we demonstrate this innovative cost-effective environmental and energy technologies. And what we're really looking to do is take the research that's come out of SERDP or other R&D efforts, and we move it into ESTCP as it matures so that we can go out into the field, collect the cost and performance data that is needed to accelerate the commercialization or the broader adoption of these new technologies. We have a couple of drivers that determine the sorts of research and demonstrations that we fund. First, we are looking at the sustainment of our testing and training ranges, as well as other facilities and operations. So that's a very broad area and can take the form of addressing unexploded ordnance, noise issues, changing environments, and maritime sustainability, just among a few. We're also looking at reducing current and future liability, and that includes the traditional sort of pollution prevention program to control life cycle costs and looking at the elimination of different chemicals of concern or hazardous materials that we may have in the manufacturing, maintenance, and operations on our installations. And then we're also dealing with impacts from past practices. And that's really looking at the emerging chemicals of concern or our legacy chemicals of concern that are impacting groundwater, soils, and sediments, as well as the unexploded ordnance liability. So we look at our program in several different focus areas, and that includes PFAS and AFFF, installation restoration, installation resilience, munitions remediation, sustainable energetics, corrosion and repair technologies, and electrical infrastructure and planning. And the presentations you'll hear today are dealing with some of the munitions constituents that we use on our installations. Technology transfer is very important to us. That is one of the driving goals, particularly under the ESTCP program, is to get out the information on the technologies that we funded so that it can be used by the end users in the community. That's taken the form of guidances and manuals. We have an environmental wiki. We do a number of different types of training, both online and in person, and then videos as well. And all of this is on our website that's listed here at the top of the page. And then key to all of this are these webinars that you're attending today. Shown here are just a few of the upcoming webinars. We generally do webinars twice a month, and you can see here that our next couple are dealing with PFAS. And on this next slide, if you go to this link, you can register now for these upcoming webinars. But also every webinar we've done in the past is archived at this site. So you can go here to get more information about past webinars that may be of interest to you. And then finally, before I turn this back to Jennifer, I'd like to mention our upcoming symposium that's going to be held in late November, early December in Arlington, Virginia area. The symposium is an excellent way to see a summary of all of the technology that we're funding under both SERDP and ESTCP. And then we also do the symposium jointly with another DoD program, OECIF and OEPF, which deals with operational energy. Every project we fund will either be giving an oral presentation or a poster. So I hope you consider attending, and information for the symposium is also up on the website. So at this point, Jennifer, I'll turn it back to you to introduce our speakers.
Wonderful. Thank you, Andrea. It's now my pleasure to introduce our first speaker, Dr. Jennifer Vedas, an associate professor of environmental engineering at the University of Utah. She conducts research and teaches in the field of environmental engineering with a particular emphasis on biological processes and the fate and transport of contaminants and microorganisms in the environment. Dr. Vedas has received an NSF Career Award in recognition of her research, teaching, and service activities. She has authored 50 peer-reviewed papers and book chapters, and she has delivered over 120 technical presentations. She received her bachelor's degree in civil engineering from Montana State University and her master's and doctoral degrees in civil and environmental engineering from the University of California, Davis. Jennifer, please proceed.
Thank you for that introduction. I'm really pleased to be here today to present a brief summary of our SERDP project on behalf of the team at University of Utah. Over the next 25 minutes, my goal is to introduce you to the nature of the problem with respect to munitions wastewater and some of the challenges with treating this unique industrial wastewater source. I'll give you a summary of some of the different treatment technologies that we think about as environmental engineers as we set out to design a wastewater treatment process. And then I'll delve specifically into our project where we looked at a series of different treatment trains, including things like ion exchange, reverse osmosis and nanomembrane filtration, and then some different treatment technologies after separation, those including some novel biological granular reactors, in addition to some chemical treatment processes such as UV hydrogen peroxide, ozone, in addition to a sort of novel approach using nanobubbles with ozone for treatment. And then I'll wrap that up by looking at the environmental impacts from these technologies by using a life cycle assessment. It's one thing to be in a laboratory and developing these really cool projects and proving it out at a pilot scale, but it really comes down to what is the cost of all those technologies compared to the baseline treatment that's currently being used, and what is the true environmental impact long-term from different technologies that you're selecting. Okay, so let's talk first about the nature of the problem. Munitions wastewater can have a variety of different munitions in it, such as new insensitive munitions, such as DNAN, NTO, or nitroguanidine, in addition to what are considered to be legacy munitions, such as RDX or others. They can be in relatively high concentrations, half solubility of some of these compounds, up to or even half that solubility. So for example, NTO is highlighted here because it is very highly soluble and also contributes a bit of issue with respect to pH. So if you have a high NTO concentration in your wastewater, your pH is actually going to be down around 3 or 4. In addition, there is water that might be added to the system that comes from a domestic source. So if that domestic source, say river water or groundwater, contains high hardness or calcium or magnesium, that might foul some of your downstream treatment processes. Okay, so high concentrations of NTO is a problem. On this next slide, I want to emphasize what some of the treatment goals might be. This is a picture from the Iowa Army Ammunition Plant MPDS permit. And I've highlighted in the lower right the concentrations of NTO that are allowed to be released on this version of the permit that goes through 2025. You'll note here that that facility is allowed to release only 1.1 milligrams per liter. So influent concentrations might be, say, 16,000 milligrams per liter, and you have to get it down to 1.1. The other challenge that faces facilities when trying to deal with these wastewaters are the volumes of water that needs to be treated. In some cases, there might be a certain batch that has some insensitive munitions as well as RDX. And then say 6 months or a year later, there may be a different batch of munitions that's being produced with a completely different constituent makeup. In addition, the volume might vary from like no wastewater up to say a few tens of thousands of liters that need to be treated. So very variable influent that is kind of an exciting challenge from an environmental engineer to design, but from an operational standpoint might be a bit of a headache. Okay, so in thinking about how we would select a technology, given all those constraints that I just mentioned, here are some of the thought process that you might go through in selecting a treatment train. So if we start on the left-hand side of this diagram, I've listed out some of the available treatment technologies categorized by, say, what would be considered a mechanical, physical, chemical, or a biological process. This is not an exhaustive list of all the types of technologies that are available, but I present some here in each category as examples. So under mechanical, you might be, for instance, trying to separate out one particular constituent that would slow down, say, a biological treatment process later on. So that's one of the examples and why we included both ion exchange and reverse osmosis in our study. You then might have a physical chemical process, say photolysis or ozonation, where you're actually now trying to target treatment of the separated munition compounds or other constituents that you're concerned about. Okay, so after you've selected a variety of technologies, you then need to screen them out and decide, yes, this is an appropriate one that I want to proceed with a lab or pilot scale. Some of the considerations you might have are shown in the upper right, where you're looking at these selection and design criteria. So one of the key characteristics is how clean does that water need to be before I can release it to my receiving water or wherever it ends up. It might even be reused within the facility, as some facilities are interested in reducing their overall water footprint, right? So whatever that level is, whether it's an MPDS limit or other standard, that's probably one of your first criteria you're going to consider. The second one might be whatever this degradation product is, or sorry, whatever the treatment technology is, it can't generate a more toxic byproduct, or it can't produce some recalcitrant compound that is not treatable before I release it. We are dealing with munitions, so obviously we don't want to have any additional explosive hazard or hazard for our workers dealing with that wastewater. It also needs to be a pretty robust technology in that it can handle a variable flow and variable concentration of these different constituents. When dealing with, say, municipal wastewater that comes from toilet flushing or sinks or showers, yes, that's a pretty high organic load, but it is pretty stable throughout a day, right? You might vary at midnight versus first thing in the morning, but you know what's coming in. It's very different when you're dealing with this munitions wastewater. And then finally, you might want to consider things like capital costs, regulatory acceptance, and one of the key ones here, number 7, I want to point out, is available operations personnel. If you have a brand new technology that comes out of the lab, you may not have workers at your facility that are familiar with the intricacies of maintaining that system. So training of these personnel at the facility is also important. So an example shown in the lower right is from Holston Army Ammunition Plant. This is just a satellite image from Google Earth of that facility, where you can see there's multiple different treatment chains involved, including, say, an equalization basin to sort of equal out that flow of 100 liters one day and 10,000 liters the next day. Various biological and chemical treatments are also used as well. Okay, so let's talk specifically about our project and what we ended up studying. This flow diagram is confusing, but I'll walk you through it. It's basically the influent comes in the orange box on the left hand, might have any one of these munition constituents in addition to things like perchlorate or nitrate, if those are also included in whatever formulation that facility is producing that day, or potentially even demilitarization wastewater. And then the effluent in the blue box on the right-hand side is our goal, right? We want to be able to either discharge that wastewater, the treated wastewater, straight to a receiving stream, or it may want to be reused within the facility. In the boxes are the different technologies that we studied. So we started by looking at different anion exchange processes, in particular because NTO was of concern to us because it may be in very high concentrations. And we also looked at a membrane filtration system to separate out, say, the larger compounds such as RDX and DNAN from the smaller permeate compounds such as NQ or NTO and perchlorate. And we then had various biological and chemical treatment processes that we evaluated for either the brine from the ion exchange backwash or the flow through or the permeate or retentate from the membrane. Okay, so let's get started. I have just a few slides on each one of these technologies. We have a series of papers that have been published. I encourage you to email me or find it through Google Scholar, and I'd be happy to share those papers with you if you need more details. So some highlights. Let's talk about the ion exchange technology. The argument for this being that NTO is an ion in most systems. It will be in very high concentration and could foul later biological treatment. So we were evaluating whether we could remove it from the wastewater and separate it from the other munitions such as DNAN, NQ, and RDX. So the plot on the lower left shows you some batch studies that were conducted with 4 different ion exchange resins. These resins were selected because they are commercially available, they're used for water treatment, and they're also specific for nitrate or perchlorate removal. What we see here in the graph on the lower left is concentration. So this is relative concentrations. A 1 being there was no removal of this munition in the ion exchange column, and a 0 being it was removed 100%. Oh, I'm sorry, the inverse of that. So let's look at NTO on the diamonds in the second plot from the right on the lower left. What we see here is that NTO was completely removed in this ion exchange resin and all the resins we tested, whereas NQ was not removed at all. DNAN was variably removed, and some amount of RDX was exchanged on this resin. We also see that in the plot on the lower right, some of the resins had a higher absorption capacity for NTO than others. So for instance, the Purolite A532E won out in most of our systems and was used in our column studies. And with respect to the column studies, we have now processed about 20 different cycles of synthetic munitions wastewater through an ion exchange column. And we've been able to successfully regenerate it with both 6% salt and also a 1% methanol hydrochloric acid brine solution. So we're able to repeatedly load that ion exchange column and remove the NTO for further treatment. And additionally, we're able to reduce the overall volume of wastewater that needs to be treated that contains NTO by about 75%. But the permeate through the ion exchange column could contain, for instance, DNAN, NQ, and RDX, which needs to go to further treatment. And then the NTO that is removed from the wastewater and now in a concentrated solution also needs treatment. So some options available for treatment of both of those streams are in the next few slides here. For IX brine regeneration, so the plot on the upper left shows you sort of the flow diagram, right? I have munitions wastewater coming in, which may require a pre-filter membrane if there are, let's say, some crystals of the munitions that have precipitated out as it flows through the treatment system because it's very high concentration. And then it would go through an ion exchange column, would be loaded with NTO within the ion exchange column, and then I'd need to regenerate it. So then treatment of that regenerate is what we're focused on here. Some of the different technologies we explored for treatment of NTO in a brine solution are shown in the figure on the lower right. Again, we're looking at relative NTO removals. And so we see that certainly both a macro scale ozone and what's called a nanoscale ozone microbubbles are both very efficient at removing NTO when I have a relatively clean water, such as deionized water. When I add in things which might be present in the brine, such as that methanol, right? Or I am adding in some complex salt solution for the regeneration, my degradation of NTO decreases, but I'm still seeing pretty effective removal of NTO. So I could theoretically reuse my ion exchange brine for regeneration of that column multiple times, which reduces the cost of the system and improves the sustainability of the system. Okay, let's talk real briefly about our other separation technology, the reverse osmosis system we looked at, and then we'll go into the biological treatment systems. On this slide here in the lower left, I am showing you for two different reverse osmosis membranes, the ROSW30 and the ROBW30, both of which were selected because they have lower energy costs associated with operating them, meaning I can get a higher flux through that system for less pressure that I have to input for that RO system to work. Those two RO membranes were very successful at separating out all of the munition constituents, with the exception of NQ. I was starting to see about an 80% rejection across when you averaged all conditions. And so what were the conditions that we tested? Well, we tested influent wastewater that was a variety of pHs, right? So wastewater could be a pH of 3, could be a pH of 7, or maybe a little higher than that. And then sometimes you might have co or counter ions also present in that wastewater. So we did a series of tests where we added calcium and magnesium and some other chlorides, for instance, and nitrate to see how that affected the rejection. So NQ, when you threw in some very complex wastewater, we started to see reduced rejection in that system. Okay, and then on the upper right, it just shows you some Gaussian process regression surfaces that we did to look at the performance of the system for DNAN and BW30, DNAN removal in a BW30 membrane. So this performance index is a measure of both the amount of flux, so the amount of water, clean water I can push through that membrane, and it's multiplied by the rejection of each particular compound. So we want a pretty high performance index, and these kinds of systems can be used at a facility to say, optimize, this is the kind of wastewater I've got coming in, this is the different conditions and performance index I can expect from the different membranes. Okay, one other quick thing I just want to mention, this is NTO and RDX diffusivity parameters. So NTO in particular, I want to point out here, because this does have modeling importance for those of you looking at groundwater transport of NTO. Hopefully it's not in the groundwater, but if it does, like we've seen for RDX, we want to be able to estimate the flow and transport of that. So we did some stop flow studies looking at NTO and other munition constituents. And what we found was if you're using theoretical models to estimate the diffusivity of NTO, you might underestimate the diffusion of NTO through porous media, whereas our experimental results showed a much higher, order of magnitude higher diffusivity of NTO. So a cautionary tale for those modelers out there. Again, this is published. I'd be happy to provide the paper to anyone who is interested in knowing more details about it. Okay, let's talk briefly about the biological treatment systems, and then we'll look at the life cycle assessments for putting all these together. Okay, so what's a granular reactor? This is sort of a new model of biological treatment systems. Most biological treatment systems are either a suspended single cell system, or you might have a flock reactor, similar to most municipal wastewater treatment plants, where there's a few bacteria that are sort of accumulated together. There's a little bit of sticky extracellular stuff that holds them together and allows them to settle relatively quickly. Whereas a granular reactor is a very tight bundle of dense microbial biomass that allows for very rapid settling and also allows for both aerobic and anaerobic or anoxic treatment within one system. And because it settles quickly, it's a smaller reactor footprint and potentially more cost effective. Okay, so an example of what the granules might look like are shown in the diagram on the left-hand side. So, the picture on the left-hand side shows two graduated cylinders. The one on the left-hand side is a standard activated sludge flock-based system that you might see in a municipal or domestic wastewater treatment system. They settle, but they take maybe an hour, half an hour to reach settling. In contrast, the graduated cylinder on the right-hand side of that figure shows you very large granules that are on the centimeter size. These things settle extremely quickly and are very dense. So the theory is, in the figure on the lower right, that you would see, for instance, NTO or nitrate on the exterior that might be treated aerobically if I'm pumping air through the system to keep the flock suspended. But on the interior of the granule, by diffusion, NTO or its degradation products would diffuse in the interior of the granule. It might then be anoxic or anaerobic conditions on the inside, and you might then continue to get anaerobic or anoxic degradation on the interior of the granule. And the degradation products would diffuse out just by chemical diffusion processes. Okay, so how did a system work? What I'm showing here is results for our aerobic granular reactor. So we had granules in there. They were pumped with a little bit of nitrogen gas for about 94 minutes, so a little anoxic condition there. Then we aerated them and kept them suspended for about 117 minutes, only a 1 minute settling time and a 2 minute draw off of the supernatant. The figures A, B, C, and D here show you the results for DNAN, NTO, RDX, and NQ. If you see a gray line, that's the influent concentrations, and then the red line are the effluent concentrations. So great removal in the system for DNAN and NTO, shown in plots A and B. RDX was a little bit of a troubling child. We ended up at about 50% removal by the end of the 700 days of operation in this system. Interestingly, we did see NQ degraded as well. The difference in the plot D on the lower left for NQ between phase 1 and phase 2 was really just a small change in the pH. When we adjusted the pH from 7 to 7.5 in phase 2 and added a little bit of alkalinity, about 200 milligrams per liter bicarbonate, we actually saw a significant NQ degradation. That actually correlated in the plot on the far lower right, plot F, where we saw that one of the NQ degradation products was actually inhibitory to these ammonia oxidizing bacteria for a brief period of time. So we see this sort of decrease in the lower right and then a pop back up. So some improvement in ammonia reduction, and then it decreased over time. Okay, a little bit about specific degradation products. I'll go over this quickly. Again, it was published in a paper. I'd be happy to provide that to anyone who has additional information or wants additional information. The take-home message is that DNAN, we did see it reduced to MENA and DAAN, and some production based off of the LCMS mass to charge ratios of these formations of these dimers that have been reported in other literature. We also saw, as has been reported in other literature, degradation of RDX down to TNX. One thing that is sort of novel about these results is we saw degradation of NQ stoichiometrically to this nitrosoguanidine. So that's a study for that's ongoing studies within our lab. We're trying to figure out if there is more mineralization of those compounds. And I point out mineralization because of the results that we see here on this plot. I apologize, these are small figures, but there's two main points I want to point out here. Okay, so DNAN, NTO, RDX, and NQ are shown in both the aerobic and anaerobic conditions. What we studied here was, could we add just carbon and nitrogen or no other carbon and nitrogen beyond what's present in the munitions, and could we get degradation? So one of the main points is under anaerobic conditions, so the 4 plots on the right-hand side, we saw significantly faster degradation of all of these munitions. So in this case, the munitions are the only thing present in the treatment systems. They weren't mixed together. So all of the degradation was faster under anaerobic conditions. The rates are shown in the blue milligrams per gram volatile suspended solids per day in each of the plots on the right-hand side. And interestingly, all of these munition constituents could serve as a sole source of carbon and nitrogen under anaerobic conditions. So very interesting, and I think worthy of additional investigation that is ongoing in my collaborators lab, Ramesh Goel. Okay, and just real briefly on these anoxic granular reactors, this is another study that we did. In this case, they were anaerobic. Interestingly, we were concerned about these granules staying together because methane could potentially have been produced in the inside or other gases that would cause the granules to break apart. And we didn't see that, which was very exciting. And we did also see significant degradation of perchlorate and nitrate in these systems that was sustained over 600 days of operation. Okay, so let's look holistically now at all these technologies that I presented. Okay, we wanted to do a life cycle assessment to step back and say, what is the real impact from these technologies, right? And so for those of you who are not familiar with a life cycle assessment, this is really doing a systematic analysis of potential environmental impacts from any product or service. In this case, we were looking specifically at munitions wastewater treatment trains. So this process allows us to do an analysis of all stages of the treatment technology, including like production of the raw materials, such as the resins or chemicals used in the reactors. We also can look at distribution or movement of those goods and services to different parts. Like, did we order something from, I don't know, somewhere in Europe and it had to be shipped to our facility in the central of the United States? We can look at the use of that system, whether it's 6 months or 10 or 15 years. And then we also consider the end of life, right? If this, as this is deconstructed and we dispose of it, what is the impact associated with that? So how did we do that? Well, we used the OpenLCA software. We purchased this EcoInvent database that has all of these different impact categories for multiple different processes or products that helped inform our life cycle assessment. And our baseline case was really comparing all of the technologies that we've looked at to granular activated carbon, which is used at some of these facilities for industrial wastewater treatment. Okay, so let's look at our plots here. The plot on the left-hand side compares granular activated carbon to ion exchange with treatment either by a granular reactor or ozone, UV, etc. And the plot on the right-hand side compares granular activated carbon to reverse osmosis. The categories along the x-axis include the various environmental impact categories from all different phases of the system. So acidification might be, is there a pH adjustment due to acid rain or release to a wastewater, for instance, from production of materials, from use of the materials, from end of life, etc. The horizontal lines basically represent how long would that technology need to be used until it had less environmental impact than granular activated carbon. So for instance, ion exchange on the left-hand side shows you that for some of the technologies, in the worst case, how long would it take until ion exchange was less environmentally impactful than granular activated carbon? In this case, it was 13 years. In the case of RO, it was 4 years. So the take-home message is if you're looking at different technologies, it's more than just the now cost for purchasing the material and putting in place. These environmental life cycle assessments are also handy for figuring out how long until the environmental costs come down to be reasonable. And you can also argue which one of these particular environmental impact categories you might be most interested in. For instance, global warming may be something you're very interested in, and you're perhaps not as interested in ozone depletion. I don't. Right. It allows you to look at different phases. Okay, so wrapping up here, we found that both IX and RO are effective at segregating these insensitive munitions from wastewater. Brine regeneration does increase the sustainability. Granular reactors are probably faster than some other flock-based biological treatment systems. And then these treatment trains in some cases do have a lower life cycle cost than granular activated carbon. So what are the benefits to DoD, right? There are multiple treatment trains that are available for IM wastewater and process water treatment. Many of these should be piloted at real facilities, take them out of the bench scale and go investigate at these facilities. And I do highly encourage a consideration both of the cost, but also the life cycle cost of all of these technologies. Okay, with that, I'd like to acknowledge my collaborator, Ramesh Goel, who did his lab did the anaerobic and aerobic and anoxic granular reactors, and then various graduate students who did the lion's share of the work in the laboratory. The munitions were provided to us from Picatinny Arsenal for use, and then we also got a little bit of extra funding from the Global Climate and Sustainability Center here at the University of Utah. And with that, I'd be happy to answer any questions.
Fantastic. Thank you, Jennifer. I'd like to remind our audience that you can type questions for Jennifer into the Q&A box on the Zoom screen. And we have received a few questions. Let's start at the end of your talk about the life cycle assessments. Which stage of the technology life had the greatest environmental impact?
Yeah, so it's an interesting question, right? Because when you're considering any technology or piece of equipment, right, we have to consider where all the materials came from and the impact from where they were manufactured, and then the transport to wherever that's used, use at that facility, and then ultimately disposal. In this case, the production of the materials was almost always the driver of the environmental risk, if you will. So for instance, ion exchange resins, the production of those resins and all of the organic chemicals that go into producing those resins drove most of the environmental impact. But that was also true for almost all of the categories, from the RO membrane synthesis or even production of granular activated carbon tended to drive the environmental impact. It's only when you had something in place for multiple years where you started to see the use began to be more of an impact.
Okay, got it. That's very interesting. Thank you. Next, with respect to the reverse osmosis treatment, were you using real munitions containing wastewater to evaluate the fouling?
Yeah, unfortunately, we were not able to obtain munitions wastewater from a facility. We were provided with a recipe to make a synthetic munitions wastewater, which we did use. One of the major concerns with reverse osmosis systems is the potential for fouling. So in our synthetic wastewater, we either started with a treated municipal wastewater, which would contain, co and counter ions like calcium, magnesium, potassium, whatnot, that could potentially scale out and foul our membrane. And we also use a synthetic wastewater recipe to add munitions to, say, a deionized water or surface water to try to model a real wastewater.
Okay, great. Once you've separated and concentrated these compounds, could a more concentrated solution then be processed using a thermal treatment system? And then that would eliminate the possibility of secondary compounds being produced. Did you consider thermal treatment?
Yeah, great question. We did not, but I can envision where a concentration method, either RO or ion exchange, could reduce the volume enough and concentrate the munitions enough, particularly NTO, where thermal might start to be attractive. The problem with some of the other munitions like RDX is they're actually very low solubility. So we're talking like tens, less than 100 milligrams per liter. So they're not going to be very concentrated in that thermal and that water treated by a thermal process.
Oh, that makes sense. Okay. And then the next question is that when you have a concentrate that has munitions and then there's also methanol and acid in the concentrate, are there any concerns with storing that that you're aware of at the facilities?
Yeah, that's an interesting question. I do not believe so. At least in the laboratory scale, there has not been. But again, I would have to do a pilot scale at a facility and think about some of the OSHA issues with respect to worker exposure and or storage of larger volumes of an acidic solution with methanol in it.
Okay.
Excellent question. Yeah, this is why we need field trials.
Sure. Yeah. Next, did you consider TNT as part of this potential mixture or as a component of the wastewater at all? And any thoughts about how that would fit in to these approaches?
Yeah, great question. TNT was not included in our mix. There's been a lot of studies done on TNT and a lot of pilot scale studies. But if I had to guess, I would anticipate that TNT would behave similarly to RDX in our anaerobic system. We would see those reduced products, and similar to what you might find in a TNT treatment in a soil that's treated anaerobically, you would probably have the reduced products adsorbing onto organic material and being retained there. So any sludge produced, say, in a bioreactor would probably have to be further treated or could be safe for disposal.
Okay, sounds good. Next, there's a question about the life cycle assessment and the assumptions used for the GAC analysis of the GAC treatment that was the baseline against which the others were compared. Do you have any details to provide about what the bases were for the life cycle assessment of GAC, how often the GAC was exchanged, or how the GAC was disposed of?
Yeah, excellent question. I'll answer the easy one first, which is the disposal of GAC was assumed to be incineration, but that was the same assumption that we made for the ion exchange resins, although the resins would be clean and probably could be disposed of as non-hazardous waste. But to equalize the two of them, we assumed incineration at the end. In terms of the assumptions about what type of GAC and how it was produced, we went straight from whatever was in the EcoInvent database, recognizing that there are many different organic material that could be used to make granular activated carbon and different methods to do that. But again, we went straight off of what was in that EcoInvent database, the latest version. Okay. But we have a paper that's coming out, and my student has an exhaustive, I must say, an exhaustive supplemental information that will have all of those details in it, which we hope will help future researchers as they're putting together these kinds of life cycle assessments.
Great. We have one last question for you, Jennifer. You talked about the granular reactor treatment. Did you give any consideration to doing a high temperature reactor that might have mesophilic microorganisms? Any thoughts about whether that could be fruitful?
Yeah, interesting question. So most, for instance, anaerobic digesters at a municipal wastewater treatment plant are run a little warmer to make them more efficient. Most of the systems that we were operating had a cycle that was only 6 to 12 hours, so it was already pretty fast. I'm sure that if we were to warm it up a little bit, you know, it doubles every 10 degrees Celsius, we might get a slightly faster rate, but we did not test increasing the temperature of the systems.
Okay. Oh, good. All right. Thank you, Jennifer, for the presentation and answering the questions. And to our audience, Jennifer will come back and join us at the end of this webinar to answer some additional questions. Thanks, Jennifer. Our second speaker today is Mr. Brian Hubbard. He presently serves as the Environmental Safety and Occupational Health Officer for Joint Program Executive Office Armaments and Ammunition at Picatinny Arsenal in New Jersey. In this role, Brian is responsible for developing strategies to mitigate environmental, safety, and occupational health risks across the armaments and ammunition lifecycle. Previously, he served as a senior engineer for environmental compliance and pollution prevention initiatives throughout the organic industrial base, working with stakeholders to transition new technologies from concept through construction and commissioning. Mr. Hubbard earned a bachelor's degree in environmental engineering and a master's degree in civil engineering, both from the University of Delaware. Brian, please proceed.
Thank you, Jennifer. Appreciate the opportunity to be here today, and I look forward to building on the momentum from Dr. Vedas' presentation. I'd like to highlight some of the outline for our talk today on the nano, iron, and hydrogen peroxide process for complete degradation of munitions constituents process wastewater. I'd like to describe a little bit of our project objectives, our site where we performed the pilot demonstration here at Picatinny Arsenal, some of the issues that Jennifer also described with the insensitive munition explosive or IMX wastewater, provide an overview of our technology as well as results from our lab and field demonstrations, and wrap up with some benefits and to the DoD and acknowledging our team. So what did we set out to do when we proposed to demonstrate the nano iron and peroxide technology? Well, first we wanted to use actual wastewater and stress test the nano iron fenton technology to the greatest extent possible. We also wanted to generate cost and performance data that we can share with project management and industrial-based stakeholders that could support the technology transition, as well as we're tracking along with the environmental regulatory limits, both states and nationally. So yeah, our ultimate goal was demonstrating and validating that our technology was an effective and rapid process for the complete destruction of those munitions constituents in production wastewater. Our site, we performed the pilot demonstration in our laboratory here at Picatinny Arsenal. We have a robust pilot system that meets operational construction requirements required for energetic and explosive production within the industrial base. We executed a batch treatment system using tank reactor 1, and we had the opportunity to dose and add in our supporting chemicals here. But what are some of the issues with the IMX wastewater? As Jennifer mentioned, there's two recently fielded within the last 15 years or so, explosive formulations. The first, IMX 101, contains dinitroanisole or DNAN, nitrotriazolone or NTO, and nitroguanidine. Second formulation contains DNAN and NTO, switches out the nitroguanidine for RDX. And these explosive fills were transitioned for mortar and artillery applications in ammunition end items. The chart on the right highlights that some of these new or emerging IMX constituents are extremely more water soluble than their historical or legacy counterparts like TNT or RDX. And from some of the earlier production campaigns and low rate initial production campaigns, our colleagues from the industrial base realized that the existing or status quo wastewater treatment technologies weren't necessarily suited for these new and novel formulations. Found out that the incoming or influent waste streams were significantly more acidic than TNT and RDX waste streams, about 3.2. On the pH scale, they were yellow in color compared with TNT wastewater, which you may be familiar with it referred as pink water. And we were also experiencing or learning about infrastructure impacts due to the low pH of the wastewater. We were seeing pumps or piping components within the wastewater conveyance systems failing at a significantly faster rate or not meeting their intended lifetime compared to when they were processing conventional waste streams. So our nano iron peroxide technology is a two-stage process. First, we introduced the nano iron to the influent waste stream. We observed, and I'll describe further in a few charts, rapid and complete munitions constituent degradation. The zero valent iron was converted to ferrous iron particles, which further spurred and catalyzed the Fenton oxidation reaction. And then from there, the ferrous ion, as part of the Fenton oxidation process, was converted to ferric. So we were using the nano iron for multiple functions within our two-stage process. To first further reduction, followed by the oxidation of the daughter products, and ultimately promote settling. So initially, we performed a market study and obtained 4 different nano iron materials. We evaluated them in an anaerobic glove box against individual munitions constituent synthetic waste streams for NTO, NQ, DNAN, and RDX. All were found to completely degrade these MCs within a 10-minute time and at a low pH of approximately 2. However, we down selected the nano first star process for use in our pilot demonstration due to ease of use and shelf life and safety and stability concerns. So from there, after we completed the anaerobic glove back studies in with 20 milliliter volumes of the wastewater, we took that, we scaled it up in the laboratory at the University of Delaware using an intermediate reactor, about 3 to 5 liters with synthetic wastewater. Once we were confident in the technology and the results there, further scaled it up in our intermediate reactor with actual IMX 104 wastewater, which is increasing the concentration by about a factor of 78 or close to 80. And then once we were satisfied with the results there, took it to the ultimate scale up to our field demonstration facility at Picatinny Arsenal. So throughout our technology development, our ESTCP project, we successfully scaled the technology by a factor of almost 1900 times or nearly 2000 times volumetrically and about 80 times concentration. So what were some of our results from our field demonstration? Over about 2 years, we conducted 7 field trial demonstrations up here at Picatinny Arsenal. Results from the first 5 demonstrations are captured here. We observed for NTO degradation by its daughter product ATO production and its subsequent degradation, rapid complete removal. The NTO completely degraded within a 10-minute reaction period, and we saw good formation of amino triazolone or ATO, and then its subsequent degradation within 60 minutes. Throughout our pilot studies, our goal was to do the complete reduction in oxidation cycles within a 60-minute cycle period, but we were seeing that everything was gone within 60 minutes. So throughout our pilot studies, we tried to stress test the technology even further and complete the reduction in oxidation cycles within a shorter time frame. And we were able to accomplish that within 30 minutes by the time of our 5th trial. So some of our RDX and DNAN degradation, similar results. Complete degradation of RDX and DNAN also within 10 minutes, good mass balance formulation, and followed by the degradation of diaminoanisole, the primary DNAN daughter product, also within about 40 minutes by the time we became more familiar with the pilot operation. And similar results from pilot trials 6 and 7, munitions constituents were gone within 10 minutes. I'm not sure what happened there in our pilot trial number 6 with the NTO, but within 30 minutes, we were not seeing any appreciable concentrations of the munitions constituents or their primary degradation products. And also in the plot on the far right-hand side, we were seeing about 2/3 degradation of the total organic carbon just throughout this 30-minute period. So even though we were tracking the complete degradation of the munition constituents and their primary intermediate products, the ultimate end product or nitrogenous end product that we were observing was ammonia. Highlighting, we did have a little bit of mechanical error with our pilot system on pilot trial number 3, so that data is kind of an outlier. But we're seeing good conversion of the munitions constituents, complete degradation to ammonia. We also evaluated the possibility of adding polymers to the wastewater after the oxidation step to see if there might be an opportunity to further enhance settling. Our team at the University of Delaware evaluated 10 polymers in total, and the best performing polymer was a now clear 8181. It did enhance the solid settling rate somewhat, but if we were going to take this technology to an industrial base facility or scale it up for full scale implementation, could certainly consider it, but maybe cost impacts with adding this additional chemical. But we also are cognizant that the nanoiron peroxide process generates solids after the Fenton oxidation reaction. So we wanted to evaluate what the likelihood, or if there's options for sludge dewatering or recommending a scale up of a belt filter press for future design and scale up considerations. We worked on some crown press tests in the laboratory at Delaware, and what they found that this crown press process was effective in separating those solid residues from our clean munitions constituent effluent. And this data could be used or provided or transitioned to engineers within the industrial base to further design or procure a sludge filter press. We also evaluated the toxicity. We did an aquatic toxicity test in the laboratory on the raw pH adjusted wastewater that contained the IMX constituents, and found that there was an improvement compared to the influent wastewater. Also, as Jennifer described earlier, we also noted that the NTO was the primary driver for causing toxicity in our IMX 104 waste streams that we evaluated, and there may be opportunities for further improving the toxicity results in the effluent of the nanoiron Fenton process by targeting NTO upfront and early. So I don't think that there's a silver bullet for evaluating these process waste streams. And some of the process issues or potential issues and concerns that we've identified throughout our ESTCP project is that we did notice there were variable munitions constituent concentrations. We were sampling from a larger reactor that was in a non-temperature controlled building, so it was subject to the atmosphere. And we'd noticed that in the winter, when we'd go to sample, the munitions constituents were lower due to precipitation or solids settling out. And in the warmer months or the summer, when the building was warmer, we'd see more of those solids dissolved and have higher influent concentrations. Something that we were also cognizant of is the source of the nano iron. It's a foreign source material, and cost to ship it to the United States could become a concern, especially with all the supply chain concerns that are taking place within the DoD and the United States recently. And then you also want to be cognizant and work with stakeholders to manage the sludge and solids that were generated as part of this process for any future scale-up considerations. However, there's multiple options that the team or the technology could take into consideration for mitigating these potential issues and concerns, such as doing additional pilot demonstrations, working with engineering design to make sure that the system or components or belt filter press meets materials of construction requirements for operation in an energetic production facility. And then, there's also could be an option to buy consumables or materials in bulk and store them until they need to be used to reduce the cost. So in conclusion, those IMX munitions constituents are much more acidic, driven by the NTO, and significantly more water soluble than the legacy explosive compounds, which does pose a challenge with applying older technologies that have been in the industrial base for 70 plus years or since World War II with those new emerging energetic formulations that have only been fielded for about 10 or 15 years. We think that it is possible to integrate the nano iron peroxide process into production footprints. It's kind of a crock pot treatment method, if you will. If you have a large enough batch reactor or batch reactor and some additional pumps to dose the consumables, then we think could be a candidate for your production process. We did demonstrate that there is also reduced toxicity compared to the influent IMX waste streams following this process. And then lastly, we'd also definitely want to be cognizant of safety concerns for transitioning this or other emerging technologies to the defense industrial base, ensuring that there's no pinch points for explosives to get trapped in pipes or flange fittings, and also make sure that it's safe for the operators as examples. Some of the benefits of this technology to the DoD that we've confirmed throughout our project, it's fast, it's very rapid. We're seeing complete degradations of high concentrations of actual waste streams within minutes, compared to some of the status quo technologies within the industrial base that could take hours or multiple production shifts to treat a large volume of wastewater. It's destructive. We're degrading those parent munition compounds into innocuous products, first their daughter products, and then to the nitrogenous products that are more amenable to subsequent treatment technologies or biological treatment before ultimate discharge, or potentially even reuse within the manufacturing process. We believe this technology is rather simple and robust. We add some iron, the acid and the peroxide, let it react for a period of 30 minutes to an hour, and we have clean water come out on the back end. And also this technology is rather scalable. We've started out at the beginning of our project honing the kinetics in an anaerobic glove box on the 20 milliliter scale. And over the course of about 2.5 years, we took it from that scale to a 10-gallon pilot reactor at Picatinny Arsenal, about 2,000 times volumetrically scale up. Acknowledgements, I'd like to thank Dr. Andrea Leeson and the support from the Environmental Restoration Technical Committee throughout our ESTCP project. They've been invaluable with sharing their insight and perspective and providing us with leads on polymers or opportunities to improve our technology and make it a more viable candidate for technology transition. This project would absolutely not be possible without the support and leadership from my co-principal investigators, Dr. Dan Cha and Dr. Pei Chu, both from the University of Delaware. We've had awesome support from Mr. Don Moore and Mr. Steve Sheets, both from our DevCom Armament Center DMEL and Environmental Technology Division, helping us get the pilot plant ready, running the pilot plant, and helping us identify those data gaps that we need to get this ready for consideration to transition to the Army or DoD industrial base. We also coordinated with Mr. Art Shattuck at Leidos as well. University of Delaware, they were the muscle and the engine behind our project in the laboratory. Dr. Kim and Dr. Zinn and Elijah and multiple undergraduate students and graduate students who had an opportunity to study and learn off of this project. And technology development, transition, material development, new ammunitions and explosives is a team sport. This project would not and could not have been accomplished without our colleagues from the DEVCOM Armament Center's DMIL and Environmental Technology Division, as well as our teammates from the Armament Center's Munition Engineering and Technology Center. So we're thankful for support from the ESTCP level, our university and academic colleagues, and Armament Center engineers and scientists as well. These are some of the resources that I referenced in my talk today. We did publish a paper on some of the laboratory studies. This is reference number 1. Be happy to provide that to anyone that's interested as well. And for more information on our project, please visit our ESTCP project page and would be happy to answer any questions. Thank you for your time and consideration.
Excellent. Thank you very much, Brian. And we do have some questions for you to answer. I'll also remind our audience you can type questions for Brian into the Q&A box in the Zoom platform. So first, Brian, we've received from multiple audience members the question about the zero valent iron nanoparticles. And where did you source them exactly? And is there any possible source in the United States?
Sure. So we sourced them from a company in the Czech Republic. We found out that they have a vendor in the United States, but we believe they're just importing it from the single point manufacturer in Czech Republic.
Okay, understood. Next, you had mentioned that there could be variable munitions constituent concentrations in the influent waste stream. What are possible options to handle that?
Sure, some possible options that we could use to maybe narrow out or smooth out the influent concentration is recommending that some of these sites incorporate an equalization basin and do these treatment as part of when their production campaign is ongoing, essentially not necessarily storing a large volume of wastewater and waiting till they reach capacity to conduct the treatment.
I see. Okay, great. Do you envision this as a standalone treatment unit, or is this something that could be part of a treatment train, more similar to some of the things that Jennifer was talking about earlier?
Sure, great question. I think it could be dependent upon the site-specific mission and need, but believe this technology could be integrated with existing treatment trains or other separation or physical chemical treatment processes.
Sure, that makes sense. Okay. What was the ratio of nanoparticles to the volume of wastewater that was required for treatment?
Sure. So for the 10-gallon production wastewater that we used here at the pilot scale, we used about 1.4 kilograms of nano iron per batch.
Okay, got it. Was there any concern about corrosion, for example, from the ferric iron, and is that why the pilot plant was constructed of stainless steel?
Sure. No, great question. The pilot plant was constructed of stainless steel because that's typically what the reactors are constructed of within the industrial base. But stainless steel material for when you're manufacturing or processing energetics or explosives, whether it's the neat material or the waste streams, the stainless steel doesn't offer an opportunity for the material to build up or stay attached to the particles. So it was more of a safety reason for using stainless steel.
I see. Okay. And then are there any other wastewaters that this technology could be applicable to?
Sure. Right now for our project through ESTCP, we've only investigated the IMX waste streams because those are what we're currently experiencing. But we also believe this technology is robust enough to handle those legacy munitions constituent waste streams, whether they're individual process waste streams or commingled waste streams containing multiple types of energetics. And then due to the nitrogenous species in the munitions constituents, we haven't investigated it, but there might be opportunities for pharmaceutical wastewater or other nitro body constituents that have a wastewater.
Okay, got it. I think one final question specifically for you. Have you thought about recycling the iron from this process? Is that a possibility?
That's a great question and something that came up while we were in the midst of executing our pilot demonstrations. I think that if we had more time, it would be something that we would look into further, but definitely something for future consideration.
Okay, sounds good. At this time, we'd like to bring Jennifer back on the line. A few questions for you, Jennifer, and then a couple for both of you. So Jennifer, you mentioned the importance of trained personnel for the treatment trains. Do you have a sense of whether personnel might be available to implement the proposed approaches? Have you considered that yet?
Yeah, great follow-on question. The reason I mention it is because if we, again, consider wastewater treatment, there are more and more water reuse technologies being implemented, right? We're now asking our wastewater treatment plant operators to also be trained in drinking water treatment processes so that wastewater is converted into something that people can safely drink. There is a corollary to what's going to go on in industrial wastewater treatment for munitions wastewater, right? If you have operators who have been working at a biological treatment system for years and years and are really experts in that system, and you now throw in, oh, now we're going to have to run an ozone system or this great technology that Brian's been working on, there is going to be some training or retraining of those operators and maybe even hiring of different people to do that work. So I don't want to discount the fact that there will be some training required for whatever technologies are implemented at new facilities.
Gotcha. Makes sense. Great thing to be aware of and plan for. And Jennifer, what do you recommend as the next step in your work? You had mentioned some field testing of these technologies and these trains.
Yeah, well, I think that's true for many of the different projects, not just mine that were funded under this call in 2019 by SERDP. It would be excellent if we could find one or more demonstration sites where all of these technologies that sort of made it through that initial gate of, yeah, this seems feasible, we should have sort of side-by-side demonstrations of them at these facilities to see which one is best for that site. And I think Brian alluded to it well in his presentation, saying you can't beat doing it at the site to really shake out some of the issues that you can't anticipate when you just do it in a laboratory at a bench scale.
That makes sense. And Brian, do you have anything to add to that about a recommended next step?
Yeah, thanks, Jennifer. And Jennifer, great question. I think that I agree for next steps would probably be beneficial to bring some of these technologies that are maturing to a facility and test them. However, that you will need to coordinate with local stakeholders at those installations and project managers who are developing and fielding those end items, whether they're mortar, artillery, or energetics or explosives, and make sure that everything's coordinated with them before we can bring a demonstration to those facilities.
Okay, thank you. Brian, stepping back a little bit, at what stage of development, given the work that you've done, would you recommend that DoD be considering the development of wastewater treatment technologies for new explosives?
Well, thanks, Jennifer. I think as the DoD is considering new explosives or energetic formulations that meet lethality and other performance requirements, as soon as those ideas are being formulated and those projects are started to be carried out at the laboratory, we should start having discussions with the folks who would need to start designing those energetic wastewater treatment process trains. Even if they're novel candidates that may not make it, the earlier that we can start the environmental safety, OC health, and industrial-based risk assessment, the better off we'll be in ensuring we can deliver that future capability to the soldier.
Got it. Jennifer, anything to add to that with respect to when to start, thinking about the treatment technologies for wastewater?
Yeah, it's an interesting question, because a lot of the considerations when developing a munition in terms of like it's safe for ecology, it's safe for human exposure, right? The parameters that make it something that will degrade if it's on testing and training range are also parameters that you might look at if you're designing a biological treatment system. You might look at photooxidation rates or biological decay rates that are based off of, how ecologically sensitive or toxic is it? So I think they're kind of hand in hand. All of those prove outs for making sure that the munition is safe for the environment and humans sort of give you an insight as to whether they might also be biologically degradable or chemically degradable. So I think they're kind of hand in hand.
Okay, that makes a lot of sense. All right, one final question. This one I think is just for Brian. How might the treatment costs for this novel treatment approach impact the use of the technology? And by that, we mean the unit treatment costs, like cost per gallon for the wastewater.
Sure, that's a good question. And thanks for that. I think that the cost per gallon should be considered in totality. And what I mean by in totality is, does this technology improve the bullets getting out the door? If wastewater treatment is a potential bottleneck, then perhaps applying the nano iron fenton technology, even though it might be slightly more expensive on a cost per gallon, could improve the speed at which those end items or material leave the facility and are delivered to wherever they need to go. I think that there might also be opportunities just on the treatment scale to maybe target some upscale or different treatment technologies by integrating the nano iron process within a larger treatment train.
Okay, understood. Thank you. At this point, I'd like to thank both of our speakers for their great presentations and work, and also answering the questions today. Thank you. Our next webinar in this series is in 2 weeks on Thursday, August 24th, and that will discuss DoD-funded research on the development and demonstration of PFAS, passive samplers. Registration is open for this and other webinars, so please visit the SERDP and ESTCP webinar webpage to register. Before we conclude, I would like to remind you that both the audio and a copy of the presentation of today's session will be on the webinar webpage in case you would like to refer to them in the future. And at this time, we would appreciate it if you could please take a moment to complete the survey that will pop up on your screen. And this concludes today's webinar. Thank you.